Okay, I'm Olof Dahlqvist Leinhard coming from AMRA. AMRA is a spin-off company from our research conducted at Linköping University in Sweden. And what I'm going to talk about today is our work to develop body composition phenotypes based on the UK Biobank imaging data in a project that we're doing together with Professor Jimmy Bell, a team at Imperial College with Professor Alexandra Blakemore. It has also been funded and in deep collaboration with Pfizer in Boston. So what we're trying to do, based on this 6 minutes in the scanner that is focusing on the whole body composition aspects. So we're measuring the visceral adipose tissue volume in red, the abdominal subcutaneous adipose tissue in blue. We're measuring the thigh muscle volumes. We're measuring the fat content in the liver. And we're measuring the muscle fat infiltration in the thighs. To make this imaging phenotype a little bit more understandable, we're working with this visual representation of this body composition profile, or BCP. So this is a 6-axis diagram where we start down here with total abdominal fat index. That's the total abdominal fat normalized with height squared, just as BMI, but without the lean tissue muscles as confounder in the metrics. Down here, we have the visceral fat index, total visceral fat volume normalized with height squared. Visceral fat relates strongly to the risk of developing diabetes and cardiovascular events, and is a key factor across the metabolic syndrome. Fat ratio, that's the ratio between fat divided by fat plus muscles. Do we have more metabolic storage as fat or more metabolically active tissue as muscles? Liver fat fraction, the concentration of fat in the liver. And that relates very much to the risk of developing diabetes. It's a debated risk factor for cardiovascular events, and for all type of liver disease, it's considered to be very important. Muscle ratio, your body weight divided by total thigh muscle volume. Do you weigh more than your body can carry, or your muscles can carry? When you lose a muscle volume, that's always a bad sign in aging and end-stage diseases. Muscle fat in the front of the thighs or the anterior thighs. Fatty replacement of muscle tissue relates to a lot of different musculoskeletal conditions, and it's also something that is seen across a wide range of diseases as a negative finding. So in this plot, you see here in green the individual body composition profile compared to, in dotted red, a healthy population mean, representing from our reference database what's a healthy body composition profile. And now we want to test this on UK Biobank and see what can we confirm the existence of a healthy phenotype. So we're looking at the first 6,000 subjects in the imaging study. We started to look at the self-reported diagnosed by doctor disease fields and looked at subjects without any metabolically relevant self-reported disease. And here we see in green the 25th to the 75th percentile of that group in this body composition profile diagram. And we see that the red star is in the middle of it, and this very much confirms the presence of a healthy body composition profile. We can look at the diabetics, comparing diabetes type 2 to controls without any form of diabetes. 279 subjects, and we see in red the diabetics, green the controls, and in brown the overlap between the groups. And we see a very exploded BCP shape. Statistics highly significant on all axes. We can build a linear model, adjusting for sex, age, and BMI, still highly significant, indicating the importance of these variables. Looking at coronary heart disease compared with controls without high blood pressure or any cardiovascular disease. 237 subjects and compared to 4,343. And again, we see an expanded BCP shape, the CHD group in red. And if we do non-parametric testing on the axis here, we see highly significant values on all the important variables at least. But if we adjust this for sex, age, and BMI, we see that visceral fat is highly significant. Liver fat is actually non-significant. Muscle fat is still significant. We confirm this by doing a match control analysis, where we for each one of the CHT subjects pick one control that is sex, age, and BMI matched. So it's not a big linear model thing anymore. And we see, again, we confirm visceral fat is significant, liver fat is non-significant. If we look down here, we see actually the mean value or median value of liver fat is somewhat lower in the CHT group compared to the control group. This is cross-sectional data. There is actually one study that 2 years ago published prospective data on similar analytics, Dallas Heart study, that followed 973 obese subjects for 9 years. And they found that high visceral fat was associated with doubled incidence of cardiovascular events. When they looked at liver fat, there was no association of the adjustment for BMI or visceral fat, meaning that the cross-sectional results we see in UK Biobank now is in agreement with prospective earlier findings. And imagine what we can do with the greater power we have in UK Biobank and the time moving forward. So to summarize this, if we start to look on the right side in this slide, we see that we have a disease-free phenotype looking like the star, cardiovascular disease and diabetes type 2, where we have differences, especially in the liver fat. But where we're heading with the research now, here you have 4 different obese male subjects with very different individual BCP shapes. The super healthy, very star-like shape, the exploded one. Here only elevated visceral fat, and here elevated muscle fat, elevated visceral fat. What's the meaning of these imbalances? That's really where we're focusing our research right now, and a lot of interesting work will come out of this. So that's basically all from me, and thank you for your work, and I want to acknowledge all the collaborators in this. Thank you.